Welcome back. Our final session for day one is Symposia on Biologics and Biosimilars. This session would explore the cutting edge landscape of biopharmaceuticals from clinical development to regulatory standards. Following our previous discussions, we turn our attention to the crucial interface between academia and industry. We have a comprehensive program lined up with 6 distinguished experts from the industry. To chair this session, I would request Professor Debashish Hota, clinical pharmacologist from AIIMS Bhubaneswar. Sir is current editor of Indian Journal of Pharmacology and he has worked in PGI Chandigarh for more than 12 years. Our next chairperson for this session is Dr. Yogendra Kumar Gupta, who is a renowned pharmacologist and former professor and head of pharmacology at AIIMS New Delhi. Dr. Gupta has contributed extensively to neuropharmacology, toxicology, and drug safety and played a key role in establishing India's pharmacovigilance framework. So I request our chairpersons to kindly take their seats on the dais. Now I would request chairpersons to introduce our first speaker, who is Dr. Shubhadeep Sinha, Senior Vice President and Medical Director for Hetero Group.
No need for introduction. I'm fine. I'll. पढ़ लेंगे?
Hello? अब आ रही है?
Yeah.
Namaskar to everybody. And if somebody has to introduce to Dr. Shubhadeep, it's a very difficult thing because कहाँ से शुरू करें, कहाँ ख़त्म करें, ये एक difficult चीज़ है. So Shubhadeep is MD Pharmacology and he is a Senior Vice President in Hetero. And his expertise is what is called as a contemporary science, biologics and biosimilars. And if I say that maybe after 10 years, majority of the drugs, majority of the diseases will be treated by biologics. And of course, it is important to know how the biosimilars are made, what are the regulatory avenues for biosimilars, because I have been dealing with biosimilars and biologics in the regulatory perspective for a couple of years now. And the cost factor is one of the major important deterrent, and that's why biosimilars are being made. And today, any pharmaceutical company which is very progressive is making biosimilars, whether for arthritis, whether for psoriasis, or whether for any immunological disease, and for cancer, and every disease will be having an answer in biologics maybe after 10 years. So I appreciate Shubhadeep's effort to create this symposium on biologics and biosimilars, and he has been able to knit very effective speakers into this session. And I compliment, and he has also supported this, I understand, this Indian Pharmacological Society conference in a big way. So I would say we should give a big hand to Dr. Shubhadeep. Over to Dr. Hota.
Thank you, sir. Dr. Shubhadeep is a basically medical doctor having done MD pharmacology degree. Now he is serving in the industry and his area is, as Professor Gupta has said, biosimilars, biologics. So biologics will be the future drugs, but now India has progressed a lot, and although we are one of the largest manufacturer of variety of biologics, only few are very successful, some are partially successful. And now what are the roadblocks and what are the advantages, disadvantages that Dr. Sinha will present, and I request Sinha specifically to cut down your presentation to 30 minutes.
That's right.
Because of constraint of time. Thank
I,
you.
तो इससे पहले कितनी देर का था?
उतना ही था सर.
1 घंटे का? नहीं, 30 minutes is good
30
enough.
minutes, yeah. So, thank you very much, Professor Y.K. Gupta sir, Professor Debashish Ota sir. It's a privilege standing here before both of you to conduct this symposium. I again feel like a student giving his viva presentation, and I always feel so when I stand in front of Professor Y.K. Gupta, whether professionally or personally. Now, why are we speaking on this? I'm going the reason, as they mentioned, that almost the top 10 of the drugs worldwide are biologics. The largest selling, in fact, the largest selling drug is also a biologic. Now, because the duration of development of biologics is very long, the costs are also very high, and that because it is becoming an essential drug in a lot of therapeutic areas, particularly in chronic life-threatening illnesses such as cancers, inflammatory diseases, immunological diseases. Biosimilars had to come where the copies of biologics, as they call, but not a true copy. That's why it's called similar. Had to come in to reduce the cost. However, as you will see that it's not that easy to even develop a biosimilar, because again the duration of development is also quite long. For this, we have speakers. We have 4 speakers. The 5th one has not come, Dr. Praful. So what I am going to give you is a brief of a brief overview of how biosimilars get developed, what are the various clinical and safety considerations, and then Dr. Shefali will take care of the bioanalytical part. Dr. Soumya will take care of the preclinical part, and the new biologics called the antibody drug conjugates will be taken care by Dr. Pankaj Thakur. So I have actually 2 sessions, the clinical and safety, because the safety speaker has also not come. So I will take some additional time. Yeah, so anyway, so I'll try to keep it. So the key aspect of biosimilars is not necessarily to re-establish the efficacy of the drug. It is to establish the safety and comparative abridged efficacy data. So, but the safety becomes a primary goal for any biosimilar to be approved by the regulatory agencies. Now, the regulatory agencies are almost like the Game of Thrones. The reason being that they all behave differently. Same guidelines, different approaches, different interpretations. So, so therefore, one study in any country will not suffice in other country. But one global study again will also not suffice in most countries, because there will be lack of sample size in each of the countries. So therefore, it becomes a very complicated task. And however, if you look at you can still divide the EU-like country, where the Australia, New Zealand, Argentina, Mexico, they all, if you have done studies in the EU, they are often accepted in these countries. The US-like country, the guidelines are similar, the US and Brazil, and in between, we don't know what is happening here. So Russia, China, India is now very well stabilized, because with the 2025 May guidelines, 2016 was also a good step in that category, but still there is a lot of unpredictability in these countries, but that's the rule of the law. Now, what are biological products? Biotherapeutics, these are active substances that are derived from biological source. Common terms, you have heard of hormones, enzymes, monoclonal antibodies, blood-derived products, and animal-derived products. But monoclonal antibodies are the targeted therapies, and that is the one where biosimilar development is also the most. Often used for chronic illnesses such as diabetes, autoimmune diseases, cancers, and certain neurological diseases, even for myocardial infarction. So what does biosimilarity mean? So definition wise, It means a version of the active substance of an already authorized reference product in the European Economic Area, where there is similarity in terms of quality, biological activity, safety and efficacy based on a comprehensive comparability exercise. Each term is important. Comprehensive comparability exercise means comparability through and through the development from the cell bank to the final, even in the phase 3 studies, everywhere there is a comparison against the reference product. In the US, it is called a follow-on biologic, which is they recognize there would be differences, but these differences should not be clinically meaningful. There can be minor differences, but they should not be clinically meaningful. India also calls it similar biologics, similar to what Europe defines it. Now coming to the most important biologics and also biosimilar is the monoclonal antibodies. These are large complex glycoproteins. So glyco and protein both are important in terms of characterization. However, this has two portions, the antigen binding portion and the FC portion, the complement binding portion, which is the effector function. These are the two important characterization endpoints. And then you have the safety endpoints where there are when the protein degrades, a lot of molecules get released. So there also characterization is required. This you have studied in the pharmacology textbook by the hybridoma technology. But so I pity my son who's studying MBBS. In our days, it was much simpler to remember the names. So we have, say, pembrolizumab. By the time you forget what the question was. Now today, so many biosimilars with biologics with such names becomes difficult. But there is a way why the nomenclature has happened. You can see there is the source, whether it is murine or human or humanized. Accordingly, you have, if it is murine, it is OMAB. If it is chimeric, it is rituximab. If it is humanized, it is trastuzumab. And if it is human, it is adalimumab. So accordingly, there are some other portions, but this is the reason why such names are there. And then there is also subsystem circulation. Rituximab we mentioned, CMAC because circulation. And that's how the naming is there. Look at the market forecast for the 10 years. Europe were the first to enter into biologics, first to enter into biosimilars. They still lead and they will continue to lead in terms of the development. And oncology diseases are the most favored in terms of biologics and biosimilar development. Monoclonal antibodies among the other biologics becomes the dominant position. And then you have oncology, and then we have among the oncology, you have the breast cancers, the solid cancer, the breast cancer is the maximum development, then you have the lung cancers, and then then you have gastric cancers, and among the liquid cancers, the blood cancers, you have CLL and NHL are the two most important therapeutic areas where biologics and biosimilars are getting developed. Biosimilars are developed after the product patent expiry. As you can see, so many products have undergone, are undergoing product patent expiry. And 2017, if you look at all the products that are there, they all have biosimilars in India as well as worldwide. Similarly, if you look at 2025, certain patents are expiring, and all the companies have started developing all these molecules. And I'm sure Professor Gupta, in his role as RCGM, you would have gone through some of these molecules, such as pertuzumab, which is one of the most favored molecule in terms of development. Denosumab, which is already first biosimilar will be launched in India, and then there are others. And if you look at the life cycle of development, a lot of time you have to find a equivalent host cell clone, cell bank clone, then fermentation, purification reaction, manufacturing scale up, and then characterization. The characterization part is the most difficult and most time consuming, where the quality of the product is, so the product is the process in this case. And then you have the non-clinical and the clinical entities. So the biosimilar programs, now the one big part about biosimilars is something called as extrapolation. So usually most of these products, especially in oncology, they have multiple cancer indications. In case of biological innovators, you will have to undergo phase 3 studies in all those indications. Whereas in biosimilar, you'll have to take care of one lead indication, establish the surrogate efficacy, and then there is, based on mechanism of action and receptor, there is extrapolation to all other indications. And thereby, if you look at the development pathway, the clinical studies are minimum in biosimilars, whereby the characterization, analytical characterization is the maximum. It's a stepwise approach so that a totality of evidence is accepted by the FDA. So does the EMA. This is the guidance where this guidance was meant for an affordable biologic for the US population. But then if you look at the US approval pathways, um, there are two pathways for a biosimilar. Either you go as a new biologic altogether, called the 351A pathway, where phase 1, phase 2, phase 3, everything has to be redone. But if it go by the 351K pathway, the biosimilar pathway, then highly similar to the, you have to show the high similarity, especially in terms of analytical characterization. You have also the EMA guideline, 2015 guidelines. EMA is typical for not only an overarching guideline, but also product-specific guidelines. And you can see there are multiple guidelines, and there are guidelines on the monoclonal antibodies. And this is how the, again, the Europe defines it as, you can see the reduction in the non-clinical, reduction in the clinical, and of late, the biggest thing that has happened is the waiver of the phase 3 studies in certain molecules. Complete waiver of phase 3 studies only with phase 1. And it has happened with pembrolizumab with two companies, Sandoz and Formicon, which is one of the most important landmark events of 2025 in terms of biosimilars. And there are other relevant EMA guidelines on immunological and non-clinical. This is the 2016 biosimilar guidelines of India, and then you have to 2025. The author is right there. The May 2025 guidelines, draft guidelines. So there is full chemistry and characterization data, physicochemical characteristics, biological activity, purity and impurities. Now, microheterogeneity, which means that when the protein disintegrates, the sulfur pairings, there is deamidation, there is oxidation, there are glutamine residues that will come out during the manufacturing process. That has to be kept under control and has to be characterized vis-a-vis the reference product. It must fall within the acceptable range, and there has to be batch-to-batch consistency. There's also aggregation, there is excipients, there is syringe, all of that needs to be seen. These are glycoproteins, so glycosylation becomes extremely important because it may affect the pharmacokinetics, safety, and the efficacy. And there could be even a new patent that might come out because of the glycosylation. But most important is the safety. And then we have to characterize the protein structure. As you know, it's a multiple levels. You have up to a quaternary structure. Every structure needs to be compared against that of the innovator. So there's attributes relating to the amino acid sequence, secondary, quaternary, and tertiary structure, biological and functional activities, kinetics and thermodynamics of binding, quantitative levels of impurities, as well as stability, and then the properties of finished drug product. So all of these need to be done. Further, the target binding, target neutralization, complement dependent cytotoxicity, all of these needs to be characterized vis-a-vis that of the innovator. It's almost like a fingerprinting that is to be done against that of the innovator. Structural characterization, functional characterization, as well as activity characterization. So the critical quality attributes are defined for each such protein, and these are some of them. I'm not going to details of that, but again, then follow that with in vitro studies. These are very important, that the complement binding studies, these follow the ICH S6 guidelines. Tissue cross-reactivity studies are not necessarily required. Further, in vivo studies. In vivo studies are only required if there is no, the characterization is not complete with in vitro studies. Certain critical relevant quality attributes have not been detected in the reference product, but have been detected in the test product, or in different amounts compared to that of reference products. Further will be taken by Soumya on the toxicity studies, but then toxicity studies are still required. Again, there have been issues of waiver. Quality differences may affect the between the biosimilar and the innovator may affect the immunogenic potential and potential to cause hypersensitivity. That's therefore it is extremely important to have a highly similar product and highly comparable product. Immunogenicity assessment is not predictive what is happening in animals versus human, but it can give much needed window that there is a possibility of so therefore anti-drug antibodies are also one of the endpoints of the preclinical toxicity studies. Others which are required for new drugs such as safety, pharmacology, reproductive toxicology, mutagenicity, carcinogenicity are really not required because these have been already established for the that of the reference product. Local tolerance may be required in some cases because these are what to say the injectable preparations, and therefore some local tolerance is often required. Now this is an overall picture, but there are guidances in different countries. There may want more some additional studies, and one set of studies may not suffice for every country. Now, in case of clinical trials, biosimilar clinical trials almost is like a iceberg, the tip of the iceberg. When we do these, we do phase 1 and phase 3 studies. No phase 2 studies because no dose have to be found. Doses are already quantified, but they are in only one indication, limited data. There are and thereby this cannot predict the safety of the product in a larger population. So there could so for when we do extrapolation of indication, we have to be very careful. There might be differences in the efficacy between the indications. However, this is still allowed, and sometimes the questions are raised based on the mechanism of action. New immunogenicity data, when it is used in large population, real world studies have therefore becoming more and more important, and new safety data, new risks, and hypersensitivity nobody can predict. So it's like a tip of the iceberg when we actually get the approval of a biosimilar product, because most of the data will still needs to get generated in the real world scenario. So therefore, there is phase 1 studies, which is not really the phase 1, it is comparative PK studies. There is no single ascending dose, multiple ascending dose, doses need not be defined. Phase 3 studies, comparative clinical studies, which is again a comparative against that of the innovator, and then the phase 4, extensive phase 4 studies are required. However, no phase 2 dose finding studies are required. Equivalence and non-inferiority, both designs are acceptable. Safety studies, again, separate phase 4 efficacy and separate phase 4 safety studies are required. Pharmacokinetic studies, now depending on the product, you might conduct the phase 1 study in healthy volunteers or in patients, depending on the toxicity of the products. And however, healthy subjects are more sensitive to provide the differences between the test and the reference. In certain patients, certain biologics, you cannot do that in healthy volunteers, so you target use the patients. Because these are half-lives are very, very long, it is impossible to do phase 1 studies in a crossover manner because you cannot completely wash out the anti-drug antibodies. Thereby, usually it's a parallel design and with a standard bioequivalence criteria. Some innovators have different product in Europe and different product in the US. So you may need a 3-arm phase 1 study, 3-arm comparative PK studies, and with large sample sizes. It could be a single dose PK studies. For example, in denosumab, we would only do a single dose PK, but in certain cases where there are chronic usages, bevacizumab, pembrolizumab, tocilizumab, there are long-term therapies. Thereby, you need to do a multiple dose PK as well for steady state conditions. Certain endpoints may be characterized, certain endpoints may not be characterized. Where it is characterized, that is, for example, VGF in bevacizumab. It's an anti-angiogenesis molecule. CD20, we know that it will act there and it reduces CD19 and CD20 values. Adalimumab reduces IL-6 and TNF alpha values. If this is characterized, this is also becomes one of the primary endpoints in the phase 1 studies. Often these are based on laboratory tests. Combined PK/PD studies can be done. So parallel design is the most acceptable design. I think the route of administration, the Same route of administration should be there for biosimilar and reference innovator. So it could be either, so primarily it is the parent most sensitive route is a subcutaneous route, which is used for the PK comparison. It can also provide the potential PK differences that during the absorption phase in addition to distribution and elimination phases. And there is also also important for sensitive assessment of differences in immunogenicity in PK. So the immunogenicity anti-drug antibodies are also secondary endpoints in phase 1 studies. Dose selection, most sensitive doses are used. Now, if the study is in a patient population, we have to use the same dosage that we use in therapy. For example, in bevacizumab, the therapeutic usage is 5 to 7.5 milligram. If we were to do a PK study in patients, we have to use the same drug for PK comparison. However, if we use a healthy population, the lower dosages give better PK-PD comparison, and we might use a lower dosages. Example, even in adalimumab, the therapeutic dose is 80 milligram, but for PK studies in healthy volunteer, we use 40 milligram, a lower dosage which will have a better concentration effect relationships. This is an example of pembrolizumab where we have been told to do these studies in PK, a randomized double-blind parallel study. So there are two tumor types. There is malignant melanoma and non-Hodgkin cell, non-small cell lung cancer. Now we were told that the metastatic melanoma is more homogeneous population. Therefore, phase 1 preferred that in malignant melanoma. Now, phase 3 studies, most of these drugs are, as we mentioned, in cancers. So, survival benefits are important for when the drug is being developed for the first time. So, once survival benefit is established, for a biosimilar, it is the surrogate endpoint, which is the response rate, complete response, partial response, stable disease, etc., are the most important endpoints. Survival need not be again proved, again compared. So it is the surrogate endpoints which are there in the innovator study becomes the primary endpoint in the biosimilar study. There are various reasons why there are challenges in clinical development. Some of them have been highlighted, and those are the ones we will discuss. Then, because there are multiple indications, which indication to choose? The indication with the highest placebo-adjusted response rate. For example, adalimumab rheumatoid arthritis, it is also used for psoriatic arthritis. But rheumatoid arthritis is a better indication because it is a better placebo-adjusted response rate. In bevacizumab, again, there are multiple indications. Colorectal cancer and NSCLC are the most sensitive indication. They are chosen. Also, they are chosen based on the disease characteristics. Now, as I mentioned, bevacizumab is useful in both colorectal cancer and non-small cell lung cancer. India, colorectal cancer is better, so disease patient availability will be much better. The survival is much lesser with NSCLC, and thereby colorectal cancer becomes the most choicest indication for bevacizumab. Extrapolation is possible in multiple approved indication inside the therapeutic area. So one cancer extrapolated to another cancer. But in case of rituximab, which is approved for both cancer and rheumatoid arthritis, you probably have to do more than one study. That is a cancer study as well as a rheumatoid arthritis study. And always standard of care has to be the background therapy in case, and it is never the case that in phase 3 the drug is used as a monotherapy for comparison against the innovator. Also, primary tumors who are treatment naive tumors, they are better, provide a better efficacy and safe response compared to that of the relapsed tumors. So they are the ones which are chosen for the comparison. Now, immunogenicity again becomes very important. Most of these drugs are biological products. Definitely, they are expected to produce anti-drug antibodies. They are proteins, and the host antibodies will be directed against that therapeutic protein. There could be product-related factors, treatment-related factors, patient or disease-related factors. Mainly the This is very important because it is there in preclinical studies, phase 1 studies, as well as in phase 3 and phase 4 studies. Again, these will be taken care by Dr. Shefali. There are multiple analytical methods. Reference products, again, if there are two reference products for the same, rituximab is Rituxan in US, Mabthera in Europe, you probably have to do comparison against both the drugs in the clinical study. And although equivalence design is most preferred, but it often becomes impractical, and therefore non-inferiority trials are smaller in size, because the ultimate goal is to get the product quickly to market and at a much cost-effective way. So non-inferiority trials are smaller in size. It has to be defined well what is the non-inferiority margin. There are so because these are long-term studies, there are difficult to retain patients. Poor compliance and poor patient retention in long studies. Patient consent. Why should they use when all the innovator products are available? In India especially, it becomes very difficult to consent such patients. High placebo response rate, certainly in rheumatoid arthritis, you might get a 30% placebo response rate. So in that case, we may have to look at alternate designs. Sometimes there is a patient-reported outcomes in rheumatoid arthritis, especially when the efficacy criteria are there. ORR is also subjective based on the radiologist interpretation, and thereby there is a lot of subjectivity in the nature of outcomes are non-optimal, and lack of coherence among regulatory bodies, rapidly changing regulatory guidelines, and standard of care also changed. So when we did the rituximab study, we had a CHOP chemotherapy as the background chemotherapy. By the time it got launched, mini CHOP became the standard of care. Now what do you do in such case? So in phase 4 studies, we probably have to do additional studies with the new background therapy. Now this is very important. The EMA guidance on the tailored clinical approach on biosimilar development essentially means if there is a highly similar biosimilar with a very well characterized product, it has undergone the basic toxicology studies, it has undergone a very effective phase 1 studies, phase 3 waivers have been granted. And this is also shown in the US FDA guidance. Again, you can see that appropriately designed human pharmacokinetic study and assessment of immunogenicity may be sufficient to whether there are any clinically meaningful difference, which is the basic definition of biosimilar. And this is the recent, and this is a stream. So this has led to two companies, Sandoz and Formicon, getting phase 3 waivers in both US and Europe. And the product which would have taken another 3 years to get the first biosimilar approval has come in 2025. It was targeted in 2028. Thanks to these changes in the guidelines in the US and the Europe. This is also there in Indian guidelines, this waiver guidelines. And however, it has never happened till date that it was there in 2016 guidelines, is there in 2025 guidelines, but it has always happened that we will had to undergo a phase 3 study besides the phase 1 study. Extrapolation of indications, main thing, if the drug which is active in NSCLC has the same mechanism of action in colorectal cancer, in head and neck cancer, you will get the extrapolation based on the studies which have been done till date, analytical characterization which has been done, and thereby those extrapolation of safety data, if it is, you can rationalize that, and extrapolation immunogenicity data which if can be rationalized. So it is there even in Indian guidelines. It was there in 2016 guidelines. Is there in 2025 guidelines. It is so extrapolation is possible if there is similarity with respect to quality, non-clinical assessment, mechanism of action, involved receptors are the same. Phase 4 approval studies, phase 4 efficacy studies are required, as well as in India post-authorization safety. Post-authorization studies is unique that it combines efficacy and safety studies, so no separate. But if you follow the NDCT 2019 guidelines, you also have to do active post-market and surveillance studies besides the efficacy studies. There's also one more concept of interchangeability, and again, it means that when we have a biosimilar, can you use it interchangeably with a reference, because it's a long therapy. So the innovator may not be available. So can you interchange with the that of the innovator? For that, interchangeability studies are required, switching studies as well as the alternating studies. However, this has also been waived by the FDA. There is no more requirement for interchangeability if again the analytical characterization is appropriate, phase 1 study has been done, and these requirements there is a FDA guidance on. And in only the FDA required such interchangeability studies in the past. This year this has also been waived. Now this is the other.
Your time is up, please.
Yes, sir. So my time is up. There is also safety section of Dr. Neetu Sinha. So I'm taking that part also. Uh, yeah. Yeah. There are only 10 minutes for this. So the pharmacovigilance certain concept traceability from biologic to so when the multiple drugs are being used, the innovator, the biosimilar, there should be traceability. And so for that traceability, batch number, GVP requires batch number, brand name, and active substance with every prescription. So there could be traceability in terms of the, you can trace whether this adverse event has been caused by that of the biological product. Everything else is same in terms of pharmacovigilance, except that batch number becomes the fifth essential element in terms of the adverse reaction, minimal essential events. Labeling has to include safety data of both reference and the test product. And as you can see, if the flow chart if there is no batch number, you cannot process the case in terms of the biosimilars. Interchangeability can cause this causality assessment issues, because uncontrolled switching from originator to biosimilar needs to be appropriately taken into account while there is a causality. So you have neutropenia after the first cycle, after the third cycle, but you had an innovator, then a biosimilar, and again a second biosimilar, then you have neutropenia. We don't know where the causality have happened. So that traceability is extremely important. Immunogenicity also adds to the complexity of the safety. So way forward is antibody drug conjugates. It will be taken care by Dr. Pankaj Thakur, and I will end my lecture here in terms of the So summarizing all of it, in biosimilars, clinical studies are mandatory in all regulations across all countries, are comparative between biosimilar and innovator. Phase 1 and phase 3s are required. Again, there's an encouragement on terms of phase 3 waivers in one product, but it will be case-to-case studies from the future, and there you will see more and more instances of phase 3 waivers. Phase 4 studies are mandatory across all countries, both post-authorization efficacy and safety studies. Phase 3 studies are there in only one indication, and then extrapolation is done. Interchangeability is a US FDA specific definition, but even this has gotten waived. Immunogenicity is required from preclinical phase 1 and phase 3 studies. Safety, naming, traceability, specific approaches to case processing, PSURs, and RMPs are needed for effective pharmacovigilance, where batch number is the most important characteristic for pharmacovigilance. Thereby, it is important that the biosimilars must get developed, must get more and more companies should come forward, because now the science is more or less established how to develop a biosimilar, and various governments and regulations are encouraging waivers of phase 3 studies, waivers of preclinical studies. Thank you very much.
Thank you, Dr. Sinha, for a comprehensive presentation about biosimilar and biologics and role of India in developing and marketing it. So, since phase 2 is not required,
yes, sir.
So active PMS you have mentioned that
yes,
is to be done, and second thing is there will be no crossover study.
No crossover.
It can be only parallel because the long T half of the molecule.
Not just that, sir, you can never wash out an anti-drug antibody. You cannot ever compare.
What are the current developments in India that will be more promising in the near future that is going on?
So right now, developments on two things are happening. The new age biosimilars, pembrolizumab, pertuzumab, the PDL1 inhibitors, and nivolumab. Then you have the ADCs, antibody drug conjugates. The already one product is trastuzumab emtansine is in the market from Zydus, but there are at least 12 to 15 ADCs that are coming up. Brentuximab, vedotin, and then further. Trastuzumab, deruxtecan, those will be the most important development. Then there is bispecific antibodies, where there are one antibody will have multiple actions. Those are the things that are the most promising, and because these biologics are already there, there will be biosimilars of those products. Yes. Challenges will be the cost, but again, I'll tell you a story of rituximab. Rituximab was 1 lakh per vial. Today it is available at few thousands per vial. Because with the more usage and more biosimilar companies coming in, competition has become so crushing that the cost comes down drastically. And India will be the example how the biosimilar can be marketed at the most effective rates.
I think Shubhadeep has covered two talks in one session. Wonderful. Starting from what is the basic understanding of biosimilars, biologics and the biosimilars and the current regulations. Just one point is, in Indian scenario, the interchangeability, how we look at interchangeability, because if the two biosimilars of the same biologic is available at a two differential multiples, 50,000, 80,000, 1 lakh, and therefore people will think of interchangeability. Is it interchangeability must for doctor to prescribe, or it is the regulatory requirement only?
So I will extrapolate from what US FDA, interchangeability definition was only available in the US FDA at one point of time, because they believe that no biosimilars can have the same efficacy and safety. But over the years it has been seen that all the biosimilars have similar efficacy and similar safety. It is not the cost, it is the safety and efficacy, and interchangeability therefore had to be proven to get a biosimilar designation in the US. That was the rule. In Europe, there is similar biologic. Now it is becoming encouraging that they are not only, there is no interchangeability rule, but also phase 3 is also getting waived. They believe that the analytical characterization, if that is done properly and highly similar biological product is produced, in that case you actually do not need any clinical studies beyond a phase 1, well done phase 1 studies. So in that case it is believed that the product is a similar biologic as we define in India. So there is no question of interchangeability even in India. Yes, cost of interchangeability in terms of usage, in terms of cost, it might be different. Yeah.
Because of time
Yeah.
constraint, we have to wind up your session.
Yes.
Very motivating and interesting. So next speaker will be Neetu Sinha.
That I have taken.
Neetu Sinha stuff.
Okay.
I have covered that also.
You have covered that.
Yes.
Okay. Okay, Dr. Shefali Gupta.
Thank you, Dr. Shubhadeep. Can we request Dr. Shefali Gupta to please come on stage? And I request chairpersons to give a brief introduction of the speaker.